I'll be laying out the assays that we put in place within Scripps research to enable antiviral drug discovery against SARS-CoV-2. And so I wanted to start by giving a brief retrospective of where we were a year ago today, really, when the pandemic took hold. And the objectives in the field were to quickly identify new medicines that we could repurpose to quickly address the SARS-CoV-2 pandemic, which was growing. And so this is a very rational approach. And so many labs and institutes throughout the world took this approach, and hydroxychloroquine was one of the early repurposing candidates that created the most notoriety. And that was really reflected that by April 2020, you can see there was more than 200 clinical trials dedicated to looking at either hydroxychloroquine or chloroquine itself, involving 130,000 individuals. So really just a massive and incredible effort put around this candidate, right? We all know it ended up not being efficacious and isn't a good molecule for COVID-19. Conversely, if you look at remdesivir, which was a failed treatment for Ebola, this ended up being efficacious against SARS-CoV-2. And now is the lone FDA-approved molecule for a direct-acting antiviral. One of the limitations of remdesivir is that is IV infusion versus being orally administered. So ideally, we'd like to find something that would have more acceptance and the ability to administer more easily by having the oral drug. And that kind of brings me to the clinical pipeline. So early last year, there was a number of earlier stage compounds that were more custom designed towards SARS and even MERS that emerged as really good reposition candidates for SARS-CoV-2. Within those is EIDD-2801. This is being co-developed by Merck, and just earlier this week, there's some really promising phase 2A data that was released that indicated that this molecule is efficacious within patients. So bringing back around, what we did at Scripps Research, like the rest of the world, was do a very intense drug repurposing program. And this was funded by the Bill and Melinda Gates Foundation. And the reason one of the major reasons that we took this approach too is because that we have the reframe library. Pete discussed this library earlier today. We consider it a best-in-class drug repurposing library. This library was funded by the Bill and Melinda Gates Foundation, in particular for neglected tropical disease, with the thought that we could identify very quick drug repurposing opportunities from that library, or even great tool compounds to help understand the biology of these other pathogens. So this is an open access library. And so we sent this library out to more than 30 external partners over the past year. And those data from those screens are actually posted on reframedb.org, and you can freely access that. What we did was internally set up a high content imaging assay in our BSL-3 facility. And this was enabled by a really wonderful collaboration with Tom Rogers here at Scripps and UCSD. And so what we did was take a wild type SARS-CoV-2 and infect HeLa cells that were expressing the ACE2 receptor. And again, all this work is done in very high biocontainment levels. So BSL-3 facility that is located in Scripps research. So this really entails a lot of extra PPE and really limits the amount of people that can work on the project at one time. Despite those limitations, we very rapidly and quickly developed a high quality assay that enables us to screen thousands of compounds per week. And this was done, spearheaded by Melina Bukowski, who's a member of my team at Calibr, but also with incredible help from Tom and Nate as well. So our strength, particularly in my team at Calibr, is drug development, or I should say also assay development. And what Melina did very quickly was miniaturize this into 384 well format and convert this into a high content imaging assay. Through Tom, we were able to get a clinical isolate from the state of Washington of SARS-CoV-2, as well as gain access to convalescent patient serum. And that serum allowed us to actually visualize, to label and visualize the viral particles in the infected wells. So this assay itself runs for 24 hours, at the end of which we fix We stained so we could now see the viral particles, as you can see shown here, as well as the host nuclei, so we could understand whether there's a cytotoxic effect of our compounds also against the HeLa cells. What's really great about this assay is that it's a very high quality, and the data turnaround time is very rapid. It's about 3 days or so from the time we start, from the time we get the images and run the algorithm to quantify the reduction, if any, in the viral particles, as well as the cytotoxicity. So what I show on the far left here is just a nice way to kind of drive home that we had a very high quality assay. We see great grouping of remdesivir and apilimumab, which is a PIK5 inhibitor, as positive controls in the HeLa assay. DMSO was our negative control. That's the vehicle, so to speak, that all of our compounds are solubilized in. And pyramycin was just our positive control for cytotoxicity. It kills the HeLa cells, so it's highly active against everything. So screening reframe, there was just a little under 12,000 compounds that we screened. This led to 61 hits. When we followed up these hits in dose response, we set a pretty loose criteria, and that was we wanted EC50s less than 10 micromolar. That's a pretty low bar. We really wanted things that were nanomolar or even low nanomolar. When we further put the bar to include selectivity, so things that didn't really have any effect on the HeLa cells, so no cytotoxicity, that then reduced the number of hits of interest to 8. So just a handful of hits that we would consider potent and selective. The nice thing about the reframe library is that in addition to having access to compounds that we may not otherwise be available to us, is that we have data around these compounds that are either in clinical development or FDA approved. Sometimes we have pharmacokinetic data that informs on the exposure plasmas that we have. The route of administration is oral, IV, infusion, topical, as well as the dose that's approved for or being used, the safety margins, and so on. So our ideal candidate would be very safe, orally administered, compound with a high plasma exposure, ensuring we get good exposure to the lungs and to help treat COVID-19. I'll say this, out of the 8 compounds that we looked at, none of them really ticked all these boxes and looked immediately as, just silver bullet repurposing agents. So what we did was expand this net, and we pivoted to include a second assay, and this was a Calibr. So Calibr are derived from human lung epithelial cell line. And unlike the HeLa cells, they naturally express ACE2, along with TMPRSS2, which is a serine protease that cleaves the spike protein and allows a natural entry of the virus into the cell and release the RNA, so it could lead to propagation or replication of the virus. Now, in the HeLa cells, there's more of a non-natural entry that occurs here. There is no TMPRSS to cleave the spike protein. So instead, it relies on cathepsins present in the endosomes to do that cleavage, and then ultimately you get release of the RNA and replication of the virus. So there's very specific inhibitors such as hydroxychloroquine that work only against viruses that go through this endosomal pathway of entry. And there's other inhibitors like nifamstat that only work against TMPRSS2. So these will be very specific to the assays. Now, the compounds that really target viral replication within the cell are going to be active in both assays. So something like remdesivir that targets the RNA-dependent RNA polymerase will show very comparable activity in each. So applying this Calibr assay to the reframe library, expectedly we got a differentiated hit list from that. So again, I'll point out how alphanterin is one of the hits. This belongs to the 4-aminoquinolines. So this is from the same family as hydroxychloroquine. We see in the HeLa assay nanomolar activity. We see in the Calibr it's dead, it's inactive there. And that's not a surprise because it has a different mechanism of entry. Now, interestingly, if we look at some of these other inhibitors that we missed in the HeLa assay, essentially, now we see nanomolar or low micromolar activity for these hits within the Calibr. So between the HeLa and the Calibr assay, we're able to generate about 10 to 15 compounds of interest to move forward in a cascade. So what I provided to you is some details around the screening assays that we put in place within Scripps research. What we did to really help validate these hits and really justify whether they should move into a proof of concept in vivo model is develop a primary cell model in which we could evaluate these compounds against human bronchial epithelial cell lines. So these are differentiated in the air-liquid interface. And so this assay is very time intensive, resource intensive. It takes 6 to 8 weeks to get to this point. But when we do, once we differentiate, they develop cilia, they start producing mucus. And these are cells that you're gonna find, they're gonna be more representative of the cells you're gonna find in your lung, where we see primarily SARS-CoV-2 infection. And this assay has been quite powerful for us to help really validate and verify that compounds coming out either the HeLa or Calibr also show activity in this primary cell line as well. So that work has been led by Karen Wolf and Melanie Kirkpatrick, who are also part of our team at Calibr. In parallel, and very early on, Tom Rogers and Nate, and now John and Namir have been working very hard to develop and support the hamster efficacy model. So this model, interestingly enough, hamsters do develop COVID-19, and we can see this little chart here shown in panel B, that in the first 5 days, we see quite a bit of weight loss in these hamsters due to the SARS-CoV-2 infection. And more importantly is at the end of the assay, we could sack these hamsters, take out their lungs, process those lungs, and look at the viral load within the lungs. And for a compound here, this is EIDD 2801. This is one that just showed the favorable phase 2A output. We see it has an incredible reduction, almost no detectable virus particles at high doses. At a lower dose, it's intermediate, and in the vehicle, of course, there's no effect there. So we've been able to really leverage this in vivo model to profile the reframe compounds, as well as new compounds that we're developing in the medicinal chemistry program. What I'm pretty excited by too is some work done by John Taharo and Namir to develop a mouse model. Anyone who's worked with hamsters know they're chubby. They're much larger than mice. And so they consume a lot more compound. It's also a little bit easier to get PK from mice. So we're looking forward to including the mouse model to really supplement our drug discovery efforts. So unfortunately, even though we set this wonderful pipeline, the reframe compounds that we identified as potential repurposing opportunities have proven to be really moderate and not really compelling compounds for drug repurposing. So at this point, I think the take-home lesson that we've got from this effort is that we really need to custom design, custom tailor compounds towards SARS-CoV-2, and there isn't going to be quick wins from drug repurposing necessarily. But the beauty of having access to the reframe library and what it enabled us to do was to actually establish a robust and very collaborative drug discovery pipeline at Scripps. And so that library really represented a very rich collection of tool compounds and that allowed us to validate and set up the HeLa and Calibr screening assays, as well as a secondary assay in the HVAC model, and even move compounds into the in vivo model. As a result, what we've done internally is pivot to developing novel inhibitors targeting the viral protease and the RNA-dependent RNA polymerase as well. And we hope to have at least one preclinical candidate by the end of the year, and it'd be wonderful to be able to present on that in the future. Because of our high throughputness and really the awesomeness of the team working in BSL-3 to really execute on all these assays, we're able to support more than just our internal efforts. And we are, through the Gates Foundation, we've been supporting testing of numerous compounds from multiple big pharma partners as well. And we put those through the HeLa and even now the Calibr 3, and a select set of those compounds will go into the HBEC primary cell model as well as the in vivo model if they're justified. So at this point, we all know that vaccines have astonishingly made it to market very quickly. And so you may be asking, why is there a need for a small molecule antiviral therapy at this point? And I think, as we're all aware, there are escape mutants that will continue to emerge that will threaten the efficacy of vaccines or reduce that. And there's just going to be a segment of population that will not accept the vaccines or don't have access to the vaccines. So for me, when it comes to neglected tropical diseases or any kind of infectious disease, we always want to have a war chest of tools available to us to help prevent and treat those diseases. So vaccines are at the top for prevention. And monoclonal antibodies are a wonderful tool as well for prevention and even treatment too. And I see a very strong role for oral small molecule inhibitors to also be used either in prophylaxis or even treatment. And we could even reformulate those inhibitors into long-acting injectables or even inhaled treatments to increase efficacy or to kind of meet different TPPs within that. So with that, I'll wrap up the presentation and just quickly go to acknowledgements. What I'm trying to drive home here is that this is a highly collaborative effort across the whole institute. Calibr, great virologists at Scripps as well that have all contributed to this. You know, at Calibr, we're very good at assay development and drug discovery, but we do need viral expertise throughout the steps. And so Tom, Dennis, John, Juan Carlos, and others have all been there to help provide that to us, both on the in vitro and in vivo side. And of course, to do any kind of drug discovery, we have quite a large network and infrastructure at Calibr, both on the HTS, CMG, chemistry side, which is led by Arnav Chatterjee, and pharmacology, Sean Joseph and Ashley Woods, as well as Mitch Hull. So with that, I'll end the talk, and I think we could transition to the Q&A.